So thank you a lot to be here for this presentation. The professor that will evaluate your oral are from Unife Marta Zell and Julie Arnaud, from IPT Louis Osterbeck et Pierre-Luigi Rosina, and from Paris, Muséum d'Histoire Naturelle de Paris, François Sema et Thomas Indico. Compliments for your presentation and your written essay. Compliments also because you have taken care a lot of the formal aspect of your presentation, and you have done a very good work for the moment, and we are sure that you will do it also for the oral. So you can start.
Okay, so hello everyone. We are group 3, and we are here to present our work about how dental wear and molecular analysis can infer in the hominin diet. Please, next slide. Okay, as we can see in this slide, we have 2 ways of study. First, we have the anatomical approach that which find micro wear and micro wear. This approach allows us to do an analysis of both ways of study and know about the ingest of nutrients and the mastication stress. Then we have the molecular approach, in which we have the isotopic analysis, who allow us to know about the diet through determining the isotopic ratios. And then the DNA analysis studies the dental calculus. Next slide. Okay. In this slide, we are going to know about what dental wear is and micro wear. At first, it's considered a pathological condition, and now is regarded as a physiological mechanism to know the adaptation of the continuous masticatory stress. The most important tissue is the enamel, who is in charge to study and can allow us to know about the microtraces and microfractures that masticatory stress produce. We study and we analyze by scanning electron microscope, who allow us and can provide information about alimentary and not alimentary habits. Next slide. Thank you. Here we can see a peek about the dental microwear analysis from Galvin et al. Here we have the most significant techniques that the investigators use, and we can see that this is through ecological and dietary inference from fossil primate specimens. According to the anthropologists, it's a common and insufficient information study with these methods to have information of the feeding habits of prehistoric people. Next slide. Here we can see the scanning electron microscope. This machine can do one of the most important analyses, which is the dental microwear texture, and is commonly used to infer the aspects of hominin diet. During the dental anthropological studies, this method was generally very limited because these methods have been shown to be slow and have a very ratio of error. Next slide. Okay, for the conclusion, or a little conclusion from that scanning electron method, the problems is when the samples that need to be analyzed are from a different environmental microscopes. So in this case, it's a negative mode to use to indicate the quality of the enamel surface, and it's necessary another mode. And that's all for the microware, sorry. Next slide.
Okay, well, how Lucia say, understanding first of all the paleobiology and ecology of hominins through their diets is important to reconstruct the context of their evolutionary history, including how they respond to environmental and climatic change over time in terms of their livelihood strategies or life and nutrition practice. We see food as an element of identity within a portion of the population. Due to the attrition found in the teeth, we can distinguish these macrowares, which provide us with information on eating practice, stress symptoms, living condition, and nutrition. So, normal data wear is a dynamic, necessary, and natural process caused by the factors mainly of the wear and abrasion. The food that individuals eat usually have different physical properties, such as abrasiveness, thickness, hardness, and breathness, requiring a different chewing process. Next slide, please. So then we find pathologies that lead to attraction like bruxism and sharing very hard food. In bruxism, we see that it's caused teeth to wear down. This pathology can be due to the chewing forces that are exerted by the masseter and temporal muscles. This wear pattern can also occur by the action of thickness, the show, or grasping object with the teeth, using teeth as a third hand to clean skins, for example, and the minced meat. In crush very hard food, such as some nuts that, combined with other elements, produce the wear and the tear of the enamel. Therefore, the size and shape of the tooth reflect the selective and functional pressures on the physical properties of ingested foods, requiring different dental morphologies to break them down. The functional wear of the teeth can tell us what a tooth was actually used for. On the other hand, abrasion can be produced by dust present in the environment or elements accidentally introduced into the mouth or by the friction of exogenous materials. Next slide, please. So, to analyze dental wear, it is particularly useful to study molar wear patterns using occlusal fingerprint analysis, that's the letters OFA, derived from 3D optical topometry in resin we do in resin models. So macrowears accumulate throughout life. We can point out that this is based on the comparison of the facet areas of wear and the measurement of the direction of immersion. From this, we can describe the main movements of the dental caps through the three spatial dimensions, providing information of the development, the spatial position, and the enlargement of the wear facet. This can be carried out by means of a microtomography scan. Finally, the OFA analysis consists of 5 consecutive steps. After complying with this, we finally imported the digital data into a software called IMQP to calculate the direction of each facet. The end result of this last step is the creation of a 3-dimensional occlusal compass of the facet patterns of each tooth that contain visual information about the direction of all facets and their inclination. And this is all for macrowear.
The isotopic analysis method is based on the principle that the isotopic composition of fossil reflect that of the food consumed in life and that the food has distinct isotopic signatures. Isotopes are atoms with the same atomic number but different mass number. This difference in weight is at the origin of the different behavior following physical processes. The isotopic analysis, therefore, consists in determining the abundance of the isotopic ratios of an element respected to now values. Isotopic analysis is a relative percentage of values expressed by this formula, right, where R is the isotopic ratio measured by the spectrometer. Next slide. From the teeth, it is possible to measure the isotopic values of carbon, oxygen, nitrogen, and sulfur, both in the mineral fraction, carbonate, hydroxyapatite, and in the organic fraction, collision. Let's see, by example, the possible application and the limits. The C13 ratio is widely used because during photosynthesis, plant processes carbon in a different way. The result is a different delta C13 values. The different isotopic ratio classify the plants in a type C3, type C4, and CAM. The application of delta C13 measured in the teeth hominin can define diets that make use greater or less of photosynthesis plants C3 or C4. It's important to underline that the value delta C13 does not distinguish between the direct consumption of the plant and derived from herbivore they have eaten them. In Europe, where their environment is monoisotopic, the study of the frequency of the isotopic delta C13 value find other application. Slide. For example, the study proposed by Richards et al. Compared the value of carbon and nitrogen to establish the exploitation of a marine resource in ancient population. Since high C13 values in a monoisotopic environment refer to marine environment, and since the marine food chain, the value of delta N15 grows exponentially, the comparison between these relationships establish an exploitation of a marine resource greater on the population of the Upper Paleolithic than the Neanderthal one. Finally, the application of isotopic analysis is effective if the sample under examination can be compared with other values. In effect, the isotopic ratio is a relative value to be interpreted.
So, the dental calculus also provides information about hominin diets. So, dental calculus that you can see on the left part forms in absence of hygienic behavior or with precise diets. It comes from a microbial biofilm, which means an accumulation of microorganisms from different species. This biofilm mineralizes at the tooth surface, confining many microscopic remains. The DNA from all species present inside the mouth is susceptible of being preserved there. Moreover, dental calculus is extraordinarily rich in DNA fragments, as it can contain up to 1,000 more fragments than the bones from the same individual. All these attributes make this method a powerful tool that shows various applications. Thanks to both microbial DNA and DNA from the organisms ingested by the individual, it is possible to know about hominin diets. To obtain a DNA sequence, the molecule should be preserved in the remains studied. This is not always the case, depending on humidity and temperature. Warm and humid climates destroy DNA in only a few centuries. On the opposite, cold and dry climates help preservation, as you can see on the graph on the right side. To realize the DNA analysis from dental calculus, some steps must be followed. The calculus breaks free from the tooth with a simple pressure applied on it laterally. A particularly important step of the protocol is the sample decontamination. Indeed, contamination is a major issue in DNA sequencing, regularly preventing samples from being analyzed properly. It should be proceeded just before the DNA extraction because bacteria and fungus may develop very quickly. Decontamination is mainly done thanks to UV irradiation. Then, pulverization of the sample allows DNA extraction. Extraction begins with the sample decalcification that implies exposure to high temperatures and mixture with different chemical components. Blank controls allow researchers to know about the presence of potential contaminants in the lab. The final step, sequencing the genomes, may be done thanks to various methods. Targeted amplification searches for specific sequences already attributed to a particular species, though it only permits to find what we are searching for. Amplicon sequencing uses a gene shared by many species and sufficiently different from one group to another to provide a good taxonomic identification. It is carried out thanks to a PCR, that is an amplification of sequences more or less matching a reference sequence. These analyses are much more precise. But the precision is even higher with shotgun sequencing, a technique that does not imply the research of a specific fragment. Despite being longer, it allows identification of every species in the sample, even those not already present in the database. So here is an example of application of this method. It deals with the long-lasting debate about Neanderthals' diet. Weyrich et al. Used dental calculus from 3 different Neanderthals individuals unearthed in the site of Spy, Belgium, and El Sidran in Spain. The identification of plant pathogens and food fungus in El Sidran fossils led the authors to think that Spanish Neanderthals were mainly plant and mushroom eaters. On the opposite, the presence of woolly rhinoceros and wild mouflon in Spy's individual calculus is an indicator of a frequent meat diet. So a mathematical approach, including chimpanzees and modern humans with known diets, classified Spies and El Sidran individuals in 2 different groups. Belgian individuals belong to the frequent meat diet group, whereas Spanish Neanderthals are part of forager-gatherers with limited meat consumption. Thus, Neanderthals' diet seemed to vary depending on regions. However, we should note that quantification with this method is less precise than with isotope analysis. In addition to this, the low DNA preservation rendered this method useful only for recent periods, at least up to now. So here is a summary of the positive and negative points for each method. While dental wear provides more precise information in the part of plants ingested, isotopes describe more widely the hominin diets and DNA extraction from dental calculus at a more precise taxonomic level. But molecular analyses suffer from a limited period of application, which is less true for dental wear methods. As all these methods furnish different information on hominin diets and suffer from their own limits, a complementary use is required. Thank you for your attention.
Thank you very much to all of you. Compliments for your presentation. And also thank you to have respected absolutely the timing. Thank you very much. And so now we can start with a round of questions. Of course, you can answer to the question as you wish, okay? Who wants to start with the questions?
I can just say something about all your work, because I am the one that proposed this subject. Really congratulations for your essay, because it was really, really well done. And it demonstrates that you really have understand the purpose of the exercise, which was to work in groups. And also you made also further research, bibliographical research to implement your work. So really compliment for that, and also for your presentation, which was really clear and uniform, which was also nice to see and to hear. So I don't have so much question, because for me you have said pretty much everything. I just have a question that is kind of tricky, because it's something that, in your opinion, how and if there is some limits on each methodology that in your opinion doesn't have not been identified in the paper that you read, okay? And if in your opinion there may be some possibility to implement and to improve the methodology that you have read in the papers. Of course, maybe there is no answer on that. Maybe you don't have any idea, because you are not doing that for the moment, this methodology. But maybe you have some idea about that.
Yes, I can answer for the DNA extraction from dental calculus. I think something that is not mentioned in the example I gave for Neanderthals is that the group that are defined thanks to references with chimpanzees and modern humans, is that the precision of the precision of the not the data, but the conclusion is not very high, as we can say they eat, I don't know, 2 times a week meat, and 10 times a week seeds and so on. But so they do kind of big groups, so high meat consumption, low meat consumption, and I think this can be the precision can be increased thanks to wider database, but I think it's normal that the database is not huge, because it's a very recent discipline that started in 2011 basically. So I think with time the precision will increase, I hope, at least.
Thank you, Pierre. Someone else want to add something?
Yes, for the microwave, sorry. I see that the scanning electron microscope is a good option to study, but have a very limitation with the texture. So it's not provide to analyze very well the textures. So has a very limitations to the quality of the replication to the enamel surface. So I think it's a good choice to use, but at the same time, you need a very positive mold to use. If not, maybe the samples can have a very ratio of error. That's all.
Thank you, Lucia. Francois has written a lot of things in the chat. Maybe you can communicate by oral, or you want me to read them?
There is some noise in my area, but I suppose that we might have an evaluation from the group regarding the kind of exercise, the way you dealt with it, how to reach a convenient result. This might help us for next year. If it is not a heavy complementary task to ask you to do. Just half a page or 1 page is enough.
Okay, yes, we can do it, no problem.
Thank you very much.
It's because Francois is asking this because you have very well understood which was the aim of the exercise and what it means to work in a group. Okay, there are...
Thank you, Marta. You will give me a CTS.
Maybe. Some other questions? Thomas, you want to ask something? No? Christophe? No. Yes? No? Luis? Okay. So I have one question for you, and it's about, do you think that to know the diet of Neanderthals, and as Pierre in the last slide has underlined, the information that we have show us that some Neanderthals in different environment or in different area have different alimentary attitude. Do you think it can be one day, because for the moment is not so much, can be reflected also in the cultural production? I mean, where they was eating mushroom, they was producing, I don't know, side scraper, and where they was eating bigger animals, they was producing different lithic industry, because for the moment we cannot find any link between these two aspects. But the problem is also that we have very few examples of clear diet detected in the site. So do you think that if we continue this kind of study, we will find also a link with the cultural aspects?
I think we can hope so, but yes, as you said, the data is lacking for the diet part. Then I'm really not a specialist of lithic, but maybe some tools might be used for several tasks, and so if we consider that one tool can be used to several tasks, then maybe we can find it in sites where the diet of the hominins were different. So then maybe it would make the correlation harder to find, as one tool have multiple use.
Thank you, Pierre. Someone want to add something else? You all agree with the answer of Pierre?
Yes, I was just thinking about finding a pan in your home and a spoon for la pasta, charcoal in Pierluigi's room for roasted meat, and a rice cooker in Dama's room.
Spoon for the pasta is not possible, okay?
Yes, I think in this case it would be quite obvious to have a correlation, but this is an extreme example, right?
But I use a spoon for the pasta.
Okay, Pierluigi, go out of his room, please. Okay, so if the questions are finished, yes, and if you don't want to add something, you are allowed to. No. We thank you for your work. We make you the compliment for your work, and we wish you a very nice day. I asked the professors to stay a little just to decide the evaluation. Okay, so you can leave. Thank you very much, and I hope to see you soon in 3D and not just in a picture. Bye bye. Thank you very much.
Bye-bye. Thank you.
Thank you. Bye.
Ciao, ciao.
Come? Vai in altri puntini. No, in 3 puntini. Interrompi.
And we wish you a very nice day.